everybody. Welcome to today's seminar. We have Professor Dantus from Michigan State University as our guest speaker. She's a distinguished professor of chemistry and physics there. But before he started, he got his PhD in chemistry from Caltech, and he then worked as a postdoc at Professor Sewell's lab, who later got the Nobel Prize. He has more than 160 publications, 13 patents, started 3 companies, and he's, on top of being a professor, also president and CEO of Biophotonics Solutions, and he's here to talk today about strategies for selective single beam cars, sensing and measuring. So,
okay. Thank you, and first let me start by thanking Ariel for inviting me. I love Boulder, I love the mountains. I always say I love the mountains and I love the ocean, and that's why I'm at Michigan State University. so that I can get some work done. And I want to thank you for coming here. And when I was preparing the talk, I wasn't very quite sure, about the audience, and you probably have all sorts of different interests, and so I sprinkle a little bit of everything in the talk, but primarily I'm going to be focusing on sensing applications, and please, I hope you will feel comfortable to stop and ask me, and especially I love digressions, and while I have I don't know how many slides, but we don't need to cover them all. And then also during the question and answer time, I'll be glad to go back to any of these or show you a few other things. And and then afterwards, if you still have some question or something, feel free to send me an email. Now, as you've heard, and you can gather, I'm kind of busy, so I'm not going to get an instantaneous response, but you will get a response usually within a couple of days. Okay. So I'm going to be talking about this cars imaging, and don't worry, I'm going to say what cars, what is it that I mean, and what is the challenge that is being solved, and so on. And the work I'm going to be talking about is primarily from these students, Paul Brzezinski, Dmitry Pestov, who was a postdoc at the time. Marshall Bremer is a new student, and I'm only going to show you one data point, and I was hoping to get an email saying that it's okay for me to show the other slides that I have, but I didn't get it on time. It usually will come a day after. I was like, okay, sure, you can show that. I was like, well, too late. But okay, maybe for tomorrow. Okay, and then let me get started, and then we'll see how this goes. So I always like to start with something a little bit more general, like what are the kinds of things that we do in our group. And in our group, if I want to try to say it in as few words as possible, we became very good at compressing very short pulses. And basically, we turn something that's usually a nightmare, which is to work with 4 femtosecond pulses. And usually that means that you're spending most of the time tweaking, and you have limited amount of time when everything is working. We think that we converted that into something that you can count on every day. And so now what could you do with this? And so we have an area that focuses in biomedical imaging. And this would be kind of 2-photon microscopy and so on. And I actually threw in a slide because I know there's interest in microscopy. And also we scan the laser, and we have a mass spectrometer, and so we collect, under atmospheric condition, we collect ions from the sample, and we have done the first subcellular atmospheric mass spec imaging, and this is the only image you're going to see about that, but I can explain more about this. We work on some ultrafast machining and processing. Now, one of the interesting things when you're a scientist, I equate that to being a painter in the Renaissance. You always need to look for sponsors. And so we did some of this, we were doing great, and then we were dropped because of some politics at that agency at that time. And so right now we're not doing machining, but we were doing machining just a few, a few months ago, and I'm sure we will do some more later. And then we work a lot with ultra-fast laser optics and nonlinear optics and all sorts of things and measurements and compressions and pulse shaping, and you'll hear some of that today. And then we are doing biological mass spectrometry. And to give you an idea, when I sat down and thought, okay, what would be the utility of a super short pulse laser? I thought, why not protein sequencing? And I thought I was really stretching the imagination. And then I started talking to my colleagues, and I was talking about little molecules like benzene and so on. And they told me, Marcus, nobody in mass spectrometry is interested in that. People are interested in protein sequencing, and there are some certain challenges in protein sequencing that cannot be solved. I was like, okay, sure, we'll do that. And then as soon as it started working, I said, there's probably something that we can do to shape the pulses and only see certain peaks. And they looked at me perplexed and like, why would you do that? We want as much information as possible. I was like, okay, scratch that idea. Just blast the molecule. And so, so that's another set of projects that we're doing, but I'm not going to talk about those. And this is the only image just to show you that we do two-photon microscopy, and we can do a Z resolved as well, because two-photon microscopy has this built-in confocality. And we haven't done any of the cool PAM and STORM and STED, so for super resolution. I think that we could. I haven't had the right combination of funding a good student and the idea and all conjuring up at the same time so that we can push some of those ideas. But the interest that we are pursuing first is to do kind of multimodal imaging, so not just getting the fluorescence, but trying to get an unstained sample, try to get different types of contrasts. And in my mind, I think identifying is it cancer or not cancer would be pretty good. And we were working on, so some of these are fluorescence, and this is a combination of second harmonic generation and fluorescence. So this is a bone, and these are the already calcified fibers are blue, and then the green, this is a mouse that's all EGFP labeled, so it's all fluorescent. So you don't need to worry about whether your fluorescent stain got everywhere. You know that every cell expresses this green fluorescent protein. And then we started looking at melanoma and trying to see are shorter pulses better or worse. And while we were doing this, we saw something kind of cool. Usually you see something like photo bleaching, and so after a while of imaging, you leave this for a minute, and then you look back and you say, wow, you bleached this area. And then we found that we can also cause kind of a photo enhancement effect. And so there's a different sample, but under certain conditions, we can cause the region to be more fluorescent. And now we're in the process of seeing, is this good or bad? And is there some trade-off? Like, okay, let's make it just 2 times more fluorescent, not cause too much damage, and then get a much better image. And then what are the long-term effects on a living tissue? We don't know yet. So that's something that we're working on, and I have I hired a new postdoc to do this. That is a true biologist that can grow the samples and make sure that the cells are happy and all these things, so that we can do some meaningful science there. So what I was thinking that I would talk today about is a particular challenge. Suppose that there's something in this box that we don't know about. Okay, and well, let's say on the surface of it, or there's something on the surface of the wall that I would like to sense what's on the wall. And in principle, I can send this green laser and detect some scattering from it, and maybe I can detect what chemicals are in there. And turns out that that's one of the challenges that are posed these days. We want to know if there's any explosive. And the idea is, could we do this with these short pulses? Could we build something that is actually, that might be rugged enough that we can think of carrying in some mobile unit? And I'm not saying in your pocket, but at least in the car or something. And then the other challenge is, imagine that you have this turbine of a supersonic jet or something like that, and you want to optimize the combustion. So they need to get this three-dimensional picture where for every volume or voxel, they want to determine what is the composition and the density and the temperature. And so could you do that by sending these lasers? And so these are the kinds of challenges. And I always think that there's a challenge in terms of sensing and imaging. And until we can do single molecule identification and detection with submicron resolution for free, there will always be a challenge. So for the next foreseeable 10, 20 years, we have plenty of work to do. And not only we, but everybody in the world can explore all sorts of different alternatives. And so in particular, we're going to propose, let's use this very broad bandwidth ultra-short pulse lasers and see if we can figure out some strategies to do this sensing, and how far can we go? Can we go to the point that we can convince people that we can get useful information? Okay, so now I'm going to start, sometimes I'm going to start from way bottom, and you're going to say, we of course know that. So please don't be offended. And then sometimes I'll say some things that will go right over your head. So don't be afraid to say, can you explain? Now, those are very concrete examples. There may be other much simpler things, like I'll show you a plot and I won't explain what is the x-axis and the y-axis. So if I skip something, please interrupt me, okay? So we want to do molecular sensing, and there's all sorts of alternatives, and this is not a comprehensive list, but we can start thinking of spectroscopy, UV, visible, IR, Raman. We might look at absorption, fluorescence, photoacoustic signals, conductivity, transmittance, atomic emissions, mass spectrometry, chemical sensing. So now you have something else that tags something or reacts, or a surface that absorbs something from the gas phase, and as it absorbs it, it changes conductivity. So there's all sorts of things that we do to sense. And in particular, I'm going to focus today on a certain flavor of Raman. And so Raman, what is Raman scattering? So actually it's quite appropriate that I'm using a green laser. So as this green laser is going through, if you looked head on into the laser light, you would see these rings of different colors. Now, okay, suppose that you do this with a correct instrument, not with your eyes. And what you would find is, if you looked, let's say that there's vibrations of nitrogen, and so if you looked something like 2100 wave numbers away, you would find, and you amplify that by 10 to the 8 times the amount of photons that you're putting in, you would find a little Raman line, okay? And so this is this would be the Raman, and this would be the Stokes shift. As you can see, less energy. And then there would be the anti-Stokes, which would be a line that would go from a higher vibrational state and produce more energy than you put in. And that usually you don't observe it because at room temperature is not populated. But if the system was hot, or there are other ways that you might be able to see some line here in the anti-Stokes. But this was to introduce the terms of Stokes and anti-Stokes and Raman scattering. And Raman scattering turns out to be a linear process. And so, if I put 10 times more laser power, I get 10 times more light. It's also a spontaneous process. And okay. And now it's linear with the concentration. That's the other detail. Now, how can we enhance this Raman signal? Because 10 to the minus 8 just doesn't quite cut it, okay? Well, there's, for example, the surface-enhanced Raman scattering. And usually what you would have is like, this was discovered on a roughened surface of silver, and it's due to the plasmonic effect. So basically, you have resonators or antennas that are jiggled by the electric field of the laser, and they have a high Q, and so all of a sudden this electromagnetic field stays ringing for a while, and that enhances the Raman response that you can see, and people claim that you can see single molecules with that surface-enhanced Raman scattering. So all you would need to do is run to the wall, sprinkle it with the silver nanoparticles, and then run back and then make your measurement, and that would work. And then this has been used for sensing applications by Ziegler, for example. There's micro Raman, so just make very, very sensitive equipment and use a microscope objective and focus it, and you can do microscopy with Raman spectra. There are some applications of standoff Raman where, as you can imagine, they'll take a big, 50 millijoule YAG laser, shoot it, and then use time detectors and, cooled CCDs and so on, and try to get fairly good signals. And there is resonance Raman scattering. So if you are on resonant with an electronic absorption, then that's another way to get this electric field to stay a little bit longer in the molecule, to have a higher chance that you have this Raman scattering. And so, these are all different ways of enhancing this Raman signal. And then there are the coherent ways of enhancing the Raman signal. I don't know if you can see this, but if you combine two laser colors that match the vibrational frequency, then the difference frequency, as you add these two electric fields, you have this beat. And this beat matches the vibrational frequency, and so you have a coherent enhancement, and you have a lot more Raman response. So this is good. And then, and now your signal is proportional to the square of or at least the intensity of both lasers. And so you put more power, you get a lot more signal. So this is good. And then another approach would be to hit the molecule with a short pulse, kind of like a hammer, and you make all the vibrations go at the same time. Now, you just need to read those vibrations, and usually what you do is you send a second pulse that's not very short, that's a little long, and it's modulated by all these vibrations, and you get back a spectrum, okay? And basically, a lot of the results I'm going to show are along these lines. And then I'm going to show towards the end how we can make out of a short pulse something that does this and then drives specific vibrations. Okay? So so when cars was introduced, they found that you could have this coherent stimulated vibration. So here are the two lasers. And then you can use a third laser to read this information, and then you would have this coherent photon with the signal. Now, what's cool is you can cross two beams like this, and there's the creation of a third beam. And these beams are directional, and they're coherent, and that's really good because all of your signal is gathered in a tight little beam like a laser. And so this is very good, and this was CARS was proposed for sensing applications, especially like for sensing anthrax appropriately in 2002, after the 2001 9/11 stuff. So since then, there's many other flavors of CARS. There's something called multiplex CARS. So here we have two single frequency lasers. In multiplex CARS, at least one of the lasers is broad bandwidth, and so you can excite more than one vibration at a time. And then there's the idea of single beam CARS microscopy. So basically, you can take a broad bandwidth laser and you can do the pump, the Stokes, the probe, all with one laser. Okay? And this was first applied to microscopy. And when we came into the field, we said, aha, we'll never get two beams to cross at their sample at the right place at the right time if we're going 50 meters away. So we can forget about that. But the single beam idea was a really good idea. Now, this usually you would do it for a microscope. You usually can't put more than 10 milliwatts on a living cell or you kill it. And so, but if you are sending the beam 50 meters away, you would need to amplify, get a lot more photons, and then send them off. And so this idea was good, but you needed to bring it up to to an amplified laser, and you needed to amplify enough for like 2,000 wave number bandwidths and stuff like that. So there were a few things that needed to be done. So we're going to proposed to use the single beam cars for standoff detection because it's a single beam, because the signal is coherent, because it provides vibrational signature for identification. So in those days, actually, our army has chose LIBS because it's a great way of getting this sensing. And with laser-induced breakdown spectroscopy, you only get atomic emission. So they can tell you how much carbon, oxygen, and nitrogen there is in the molecule. But there are explosives without carbon, and there are some without oxygen, and there are some without nitrogen. So you want to have vibrational signatures. Then you know exactly what you have. And that's very important. It provides, then can be used to determine temperature and concentration if you need to. So this is what we picked. And so the idea is you'll have some kind of a spectrum, and of course, ideally, you would have like a square spectrum that spans everywhere you want. In this case, we picked the way we were generating the bandwidth, and we still do, is using a hollow core fiber that doesn't give you a perfectly smooth spectrum, but that doesn't matter. And so we take this entire bandwidth, does the pump and Stokes, and then we have the probe, which is a small portion of it. And then there are all sorts of tricks that have come from various different groups, primarily Silverberg and Leone, where you can put a pi phase, you can rotate polarization. There's all sorts of tricks that you can do to enhance your signal. And then you measure the cars, which is not shown to scale. Okay, so this is the laser, and the car signal is quite a bit weaker. So we started working on this. We presented our first results in Clio 2007, then we published it, and then a few months later, the group from Silverberg also used a very similar approach, interestingly at the same distance and so on. So I'm going to show you some results of what we have. Now, before we do that, a lot of what I'm going to show you depends on how you manipulate the laser. So when I'm showing you this, I'm showing you a laser that has, let's say from 760 to 860. So you're manipulating 100 nanometers of bandwidth, and you want to make sure that it's either all of it in phase, or you want to determine what phases you're adding to the different bandwidth. You're changing, you're putting a phase step, you're changing polarizations. How do you do all that? And besides, that doesn't matter where you buy your laser. As far as I know, nobody sells you a perfect laser. Especially out of the box, the laser doesn't know where it's shooting, right? And dispersion in air is also very significant. So you need to sense all this dispersion and everything to make everything perfect. Then you can do the experiment. And so for that, what we're going to use is a pulse shaper. And I think, to my knowledge, this is the first drawing of a pulse shaper. This is from a drawing of Freuly. And the idea is that you come in, there's a grating, you disperse the beam, you collimate it, and now you have a Fourier plane where you can manipulate the amplitude and phase, or the phase and the polarization, and then you can reconstitute the beam. And in a sense, now you have a handle on your system. Now, to give you an idea of how this works, I have actually, I'm going to show you a simulation program. And by the way, as Ariel said, I am the president and CEO of the company, so I'm biased for this company. So you should take this, at least with full knowledge and disclosure. So if this is a pulse shaper, and we have here the spectrum of the laser, and this is the phase, and this is the electromagnetic field, and green is a transform limited pulse as a reference, okay? And this is the second harmonic signal. And we can say, okay, what happens if we change? So if we expand the phase across the spectrum into a polynomial, so we have phi 0, and then the fundamental phase, then we have a first order, a second order, and so on. If I change the first order phase, what you see is, in a sense, it's a delay of the pulse. So once you put a pulse shaper in your laser, you don't need moving parts to delay the pulse. And then you can change the second order phase, for example, And as you can see, this is also known as linear chirp, and it broadens the pulse. Okay? And then you can have high order dispersion. For example, this is cubic dispersion. And cubic dispersion causes something kind of cool, which is that you now have more, you have the same amount of signal that you used to have for a transform limited pulse, but you can suppress second harmonic generation elsewhere. And that can be kind of useful, for example, if you're doing two-photon microscopy, because you have excitation at whatever wavelength you want. And then if I do this, see, you can tune where you get two-photon excitation, for example. And you can have, for example, some other cubic and fourth order. For example, some fiber lasers will have quartic and higher order dispersion, which are very difficult to measure because this is this low-level platform, okay, that is hard to detect. And then now, what we're talking about is stimulated Raman, and so is there any way to cause some selectivity with stimulated Raman? And so if we look at a stimulated Raman from a femtosecond laser, you would have the entire bandwidth. So basically, we're going one photon up, one photon down. And then you could try a few of these things, but they're not very good. So this just loses your signal. Cubic also loses your signal. Fourth order also loses your signal. So it's not trivial, but turns out that sine function works quite well. Okay, so as you can see, now I have selective excitation that I can tune. Okay, and it works really well, except for the fact that you start getting the second harmonic and third harmonic of these sine functions. And it's not very clean, and I'm going to show you later how this can be cleaned up. So this is how we're going to use the phase modulation to get selective excitation in cars. And I'll explain why that is important if you want to do cars imaging. So now, let's get back to the presentation. Okay, and we can always go back to the pulse shaper. Okay, so the idea is we have femtosecond oscillator. We actually have two shapers, and that's an advantage that I have because I have access to a lot of pulse shapers. And in this case, this one optimizes the entry to this hollow waveguide. So we have amplified shaped pulses coming in, and then we have a second pulse shaper that takes the whole bandwidth and it's compressed to the target. In this case, we had 12 meters after bending folding the beam a few times on the table. And the idea is these peaks are the signal that we're looking for in cars. And this is when all the phases are corrected. Now, if you don't correct for the phase distortions, this is the level of signal. And maybe we see this one, but there's nothing else here. And OK, so we wanted to get everything we want to. It's important that we correct for the dispersion. And in this case, this for a 10 femtosecond pulse. As a function of distance, it gets dispersed in air. If it was 5 femtoseconds, it would be much worse. So a 5 femtosecond pulse becomes 12 femtoseconds after a meter in air. And the signal, because it's a third order process for cars, your signal will go way down. So by the time you go to 50 meters, you lost everything. But the thing is, if you have a shaper and you know what the distance is, which you can easily measure through kind of LiDAR, then you can correct this, you can pre-compensate, and you can get the pulses exactly right at the target. And so the system we're using is a typical regen system, kind of the old positive light regen design. And then it goes to the hollow waveguide, which is here, and then comes out. And we have a grating here just to show you that the beam has a really, really broad bandwidth. You get the full rainbow. And we're picking just a small section of it. Everything else we're throwing out. And so here you have in logarithmic scale, kind of like when we use the entire bandwidth, this is more or less how it looks. And we're using here a silicon detector. So primarily here we have the detector that is no longer responsive. You have this kind of spectrum. Here it's cut off by the pulse shaper. And we can measure the phase distortions across this, we can compress them, and you can see that the second harmonic now spans from like 325 to almost 500 nanometers, so really broad, and this, in this case, is like a 4.8 femtosecond pulse. Okay, so one of the things do any of you work with femtosecond lasers? Yes? Okay, so I'm going to show you how we measure our pulses. I'll do this very briefly, and I can talk a lot more about this, maybe after the presentation. And suppose that we have a certain phase that's distorting our pulse, and we don't know what it is, and we would like to measure it. It turns out, but I just show you with the pulse shaper that the linear phase just causes a delay, and that's not what we want to know. What we want to know is if there's any curvature in the phase, because the curvature is what's going to broaden our pulse. And if we want to measure curvature from math, you know that you want to measure at least the first derivative, but even better to measure the second derivative. If you can measure the second derivative, you got the curvature right on. So let's look at the second derivative space, this unknown phase distortion. And our challenge is, can we map it in some way? Okay? So what we need to find is some experimental way to finding these green dots. Turns out that, what this plot is telling you is, I wish there was a way for me to introduce these constant dashed lines, okay? And then just find where they cross the line, right? Well, these dashed lines are constants in the second derivative space. If I integrate a constant, I get integrated once, I get a dependence on omega. I integrate twice, I get omega squared. Well, omega squared dependence, that's linear chirp. That's just a quadratic phase. Well, that's really easy to introduce with a laser, and especially easy with a shaper, right? I mean, I just show you how easy it is to just, move the thing. And so I can introduce a reference phase that will be a constant chirp, okay? And at some point, that minus the phase that you don't know will be 0. That means somehow the curvature of one phase minus the curvature of the other, at certain point in the frequency domain, they're going to cancel each other. Where they cancel, the local chirp is 0. Now all we need to do is think how can we go to the lab and find some response that would be maximized when the local chirp is 0. Turns out that if we're doing some frequency generation in a crystal, the maximum is going to be where their local chirp is 0. So we're going to take second harmonic spectra. Okay, so here's you put a thin Dublin crystal, and you scan this linear chirp, and you get this feature. Okay, and then how do I reconstruct this from this? All I need to do is a contour map as a function of this linear chirp. So I do this. I put this second harmonics in a contour map, and I get a direct measurement of the second derivative of the phase without any retrieval algorithm, without any magic or anything. It just directly comes out. Okay? Now, this is great. And now, if you did it on your laser, which, by the way, if you have an amplified laser, all you need to do is move the grading spacing. So it's an experiment that you can do immediately today. Or if you have an oscillator and you have some prism compensator, you can just move the prisms, although prisms do introduce a little bit of cubic phase. And so this is great, but this is like the check engine light. Okay? It's just telling you that your laser is not what you wanted it to be. It has some high order dispersion. Yes, but if you did this with a pulse shaper, all you need to do is put minus the integral of this phase, and then you get transform limited pulses. And so this is a 5 femtosecond pulse that had some 500 femtosecond cube third order dispersion, and this is the measurement. Okay? Now, when we sent this, we were really excited. The reviewer said, this sounds really, really good, but this data is too good to be true. Can you show us some other arbitrary phases that you can measure? And we, of course, agreed with the reviewer, and then we sent a few other phases. But this is just to show you that exactly what we put in is what we measured. Now, this is what we call in our lab MIPS2, and we had an earlier version of MIPS that uses sine functions as a reference, and sine functions have an advantage over these functions in certain cases. And so to tell you the truth, this student was able to first correct the phase distortions of the laser he was using, including the phase distortions by dielectric coatings in the mirrors in the cavity of the laser and so on. And then he was able to put pure 500 femtosecond cube, and that's why he got this beautiful line. Okay, but anyway, this method works, and to show you how well it works, we recently put a very short pulse laser that's capable of putting 4 femtosecond pulses through a high numerical aperture objective, and we were able to measure this interferometric autocorrelation, which the spectrum is kind of squarish. I mean, it's not Gaussian at all. And what you see is the black is the simulation of a 5.5 femtosecond pulse with that spectrum, and this is what we measure. So once you have a pulse shaper and you make a measurement, as I showed you, you can use the pulse shaper to make 1 pulse or 2 pulses, and you can do an interferometric autocorrelation without having to introduce beam splitters and so on. So you can make sure that you have the right thing. Okay, so how do we use this for a standoff detection? We use the entire bandwidth of the pulse. It hits the molecule like a hammer. Then we take a small portion of the beam that's longer in time, maybe 2 or 3 picoseconds, and it's modulated by this ringing, develops these sidebands, and these are the sidebands that we measure. And so here's toluene, and this is orthonitrotoluene and meta-nitrotoluene, and this is ortho-meta-para-xylene. So here's the advantage of using something that gives you vibrational signatures. So if you were measuring the amount of carbon or hydrogen, you would never be able to look at isomers and so on. And so this is 12 meters, 10 microjoule pulses, and now this is on 100 micron cuvettes, and there's a mirror behind it. So, you know, we were very honest. We wrote everything up in the paper, and then we tried some other things like vapors and solids. So here's polycarbonate and polystyrene and polymethyl methacrylate. And this is the single shot performance. This is of toluene. So in 1 pulse, you get the entire spectrum, and that's very useful. And this is, okay, so if you don't have a retroreflecting mirror and you have just some scattering surface like the wall, and then you put a drop of the liquid, now you have less than 100 microns thickness, and you have a lot more scattering. The signal is not perfect, but when you do background subtraction, you get exactly what you needed, and you can still identify different molecules. And then you can have mixtures. And now, this is how far we took this work, and then that's before politics set into that particular agency, and then we lost our funding. And we needed to, at that point I thought, well, if we keep doing good work, some other agency will be very interested in this, and we did find 2 agencies that are interested. So unfortunately, one of the agencies doesn't like me to, well, I need to send everything I need to show like 45 days in advance, and I don't usually do that. So I'm only able to show you this slide. But we're already detecting kind of like nanograms per centimeter squared. This is on a 4 micrometer thin, so 4 micrometers, polystyrene film. And we can do this, and we can do all sorts of other really cool stuff, but I didn't get the email that I can show you this other cool stuff. But I can show you the stuff from the other agency. Okay, but before I do that, in these cases, you get 1 pulse and you get the entire spectrum. Now, if I can get just intensity, or if I can get a full spectrum, I will always opt for the full spectrum. And that's great. However, if I want to obtain images, okay? I don't want to measure just what's on this spot. Maybe I want to measure what's in the room. You know, I want to scan the laser. I'm not going to do that. And I want to know what's there. Then you can, of course you can take a spectrum per pixel. Of course you can. But it's just a lot more work, okay? What would be cool is if you can train your laser and say, train it on, glucose or whatever you want, any molecule, and then you can scan, and then you know what it is. And for that, we need to learn how to train the laser. So we go back here. We say, okay, how are we going to optimize it? We have the phase and the amplitude. What should we do with it? And we have the advantage that we start from 0, because we have the device that will measure all the phase distortions and make them 0. So that's an advantage. But now, these are too many parameters. And so, even if we have, let's say, 100 pixels, and we have so for n pixel, n bit shaper, we have 2 to the n times 2 to the n times to the nth power, or like 10 to the 600 things to try. That's a big number, okay? So, and it's not like the number of grains of sand in the ocean. It's estimated that the number of atoms in the universe is about 10 to the 79. So 10 to the 600, you're not going to graduate anytime soon, okay? So, so you need to figure out really much more clever ways and faster ways of doing this, and I'm going to show you. And these genetic algorithms have been used successfully to get to these vibrations. The thing is, you can't just do an entire random search. You need to somehow, for example, if you know that sine functions are good, you can say, okay, modify these 3 parameters out of the sine function. Then you can do that within a couple of days, no problem, or even hours, okay? So these things can be done, and the sine function was used by Silverberg, and then you can use pairs of chirp pulses. That's a wonderful idea, and this was done by Mernon and Captain, showed works great, and then there's many others, okay? And then how does this work? So the sine function, going back to the program, you have this sine function, and in this case, if this is the spectrum, this is how the electromagnetic field looks like. And in a sense, if you see in the time domain, you break the pulse. The one pulse is now broken into a multiple set of pulses that match the frequency of the molecule, of the process you want to drive. If you remember in the very beginning with the cartoon that had this 2-color laser and that there was some beat note that was moving, this is exactly what you're trying to do. And you get this selectivity. Unfortunately, you get a bunch of garbage here that's not very good. But turns out that if you use this pair of chirp pulses, which you can do without breaking the pulse into 2 or anything, you can just do this phase, you get really nice sequences of pulses, and this is a lot cleaner. This works very well. And now I'm going to show you a little bit with binary phases, and I think I'm doing okay. So here's what you had to work with. Now, if you forget about the amplitude and you only use binary phases, then you cut down the search space tremendously. Why? Because you have e to the I0 and e to the I pi, and you want it to cause maximum constructive interference at one frequency and maximum destructive interference everywhere else. And turns out that that problem has been solved. In fact, it's used by GPS location and by all sorts of other measurement techniques. And so you can use these special non-recurring or pseudo-random binary phases, and you can get these solutions. And so you can get spectra like that. This in second harmonic. Oops, that's the problem with some of the green laser. And so let's say that we wanted to measure integrated second harmonic, and we wanted to measure whether something is good or bad. Then we would have the binary phase, and then if we're measuring integrated second harmonic generation, then transform limited pulses should give you the maximum. So this one corresponds to 0, 0, 0, 0, this one to 1, 1, 1, 1, 1 all across. And so this is a good way of mapping whether we're doing well or not. And now, for stimulated Raman, each of these pixels is a measurement. Now, we go to the lab and we say, okay, we want to excite a particular Raman shift, okay? And so we determine that that's the window of good, and all the background is not good, okay? And then we do the division, and then we do the theory, and so it turns out that for any n photon process, the equations have been written down, and so you can simulate this, and you can go to the computer, and you can simulate for a 16 pixel phase. You can simulate the entire experiment, and that's easy to show. And so we wrote the paper, and we said, well, actually, we know the solution. And the reviewer said, we don't believe that you have the solution. And so I convinced my students that that we needed to do the experiment. And I said, we'll have to do it once. That's the good news. The bad news is for 16 bits, you need to do 65,536 measurements. And the students were correct to point out that there's quite a bit of symmetry here, so maybe we just take one quadrant or something. And I said, look, guys, let's just do it once. I know it's painful. And so my grad student, Bingwei Zhu, and my undergraduate, Janelle Shane, went to the lab and did all these experiments. And this is what we got. And this was really pleasing, and we said, okay. So we wrote a second paper. So this was an Optics Express paper to support the PR. It was PRL. It moved to PRA paper that got published. But now it showed that our solutions that we were proposing were correct, and this shows that now we have done all the experiments. We know that there are very many solutions that are close, so there's not one solution, and there are many ways to solve this. And now that we know that our theory is fine, we can then take results from GPS that are not for 16 bits, but 203 bits. And we just write the solution, and we don't need to do millions and millions of experiments. So here's how this works with binary phases. If you have a square spectrum and you put this binary phase, you end up with this very nice clean spectrum. And now the thing is, okay, how does this work in reality? And I'll show you a few figures and then show you some other things. So this is selective excitation of vibrational modes of methylxylene at 12 meters, and you can selectively excite either this mode or this mode. And usually when you have a neat molecule in solution, there's going to be a lot of energy transfer and so on. So I was impressed that we could do this well in solution. Now that we did this remotely, it was extra. And so this is how the different phases stack up and how they look. And this would be kind of like what an X-frog measurement would look like. X-frog or yeah. And then we can simulate theory and experiment. So this is if we have like a sine function or cosine function. This is what the theory says. This is what our experiment. This is the non-resonant part. And then when we put toluene, you can see that there's some interesting things like these loops and stuff. But it works, and we can get selective excitation in these two modes of toluene. And we have done a few more tests of different things for gas phase and excited and solids and liquids. Let me show you some results from air. So this is with the spectral energies and the Air Force, and they're interested in combustion. So we can do selective excitation, and here we're showing for CO2. CO2 turns out to have this Fermi dyad. So CO2 is a linear molecule, has a symmetric stretch, but turns out that there's a bend. And if you put two quanta of the bend, it looks like a symmetric stretch. And so there are these two lines that are essentially at the same frequency, and we can excite one or the other one, and if we excite in between the two, we see nothing. And so we can make a very quick CO2 sensor, but this sensor can be for any wavelength. And then we just need to detect intensity. Okay, so we can train now. We now know exactly how to tell the laser to look at whatever molecule we want. And so to show this, suppose that there's some gas coming out of this jet, and we want to know what it is, and then we want to make an image of it. And then this is the picture, and we're going to image this area. And then we transform limited pulses, and now we're detecting with the integrated, so we don't have a spectrum per pixel, we're just collecting the intensity of the beam. And so with transform limited pulses, we don't see anything. But then we go to this one vibrational mode, the symmetric stretch of CO2, and now we can see the CO2 and we can see this turbulence. And this was actually an added bonus, the turbulence. So we scanned the beam horizontally and vertically, and we made sure that it's always there. And then I talked to an expert in fluid motion, and he said, oh yeah, I bet you that your aperture was not circular, and so if it's rectangular, this is exactly what it's supposed to do. And so I was like, okay, great. So we can do this type of imaging, and I was going to show you cars imaging on films and stuff like that, but I can't. But we can do this in gases and liquids and gases, liquids, and solids. And the last thing I wanted to say is, now, okay, so you saw the setup for this system. It would hardly fit on this table, okay? And it's kind of floating, and we're in the basement of a building, and it's all temperature control, and it's expensive. So how can you make this rugged so that we could use it, in some mobile unit? Well, you may have to use something like fiber lasers that are usually rugged. At least they don't have many adjustable parameters. And so there are groups that have shown how you can do these cars or single beam cars with fiber lasers, and there are ways to get the bandwidth and so on. And so we have worked with Toptica on this what they call their FFS laser. So it's a fiber laser, and it has a PCF fiber, and then we put our pulse shaper, we measure at the target, and this is the spectrum before compensation and after compensation, and this is time. I should put here time. I don't know if you can see yeah. So we compress this to about 12 femtosecond pulses out of an erbium. This was an erbium fiber laser, and this is the second harmonic generation before and after it was compressed. And then this is an ytterbium laser, not fiber, that goes through a PCF fiber, and then the spectrum is all messed up, and it has this highly nonlinear phase on it, and then we correct it to get very short pulses. And so now we can go to solid state, very stable lasers, use one of these highly nonlinear methods to get supercontinuum, compress it, and then we can do all of these sensing applications. So this is getting me to the end of the talk. We have a number of collaborations, and one of the exciting things is that we made these sources so user-friendly that in my group I always have something like 3 undergraduates working and a number of graduate students and postdocs, and we also have collaborations, and we have people coming in. We have once a year we have a workshop. This time we had 40 people coming in. And I can plan, sure, you'll get to interact for 4 hours with the lasers and use them hands-on, and that's the difference between a conference and a workshop. And I know that I can tell my students, run the oscillator that runs at 4 femtoseconds, run the amplified laser system, let's run it at 4 femtoseconds, and let's have the other laser systems, the amplified and the amplified shape pulses and the microscopy, for everybody to interact with. And I don't need to worry whether the laser is going to be working or not, or if they're going to be able to, if someone's going to have to tweak it, because we don't tweak the lasers. It's all done without opening any boxes, and that has been really, really useful. We have this very good interaction with the Air Force and with Sukesh Roy from Spectral Energies. We have a collaboration with Sonny Shi where we're using this for microscopy. So he has been doing a lot of cars microscopy, and we help him with a polarization shaper that he's using for distinguishing biological samples where the cars' signatures have a high degree of overlap, and I could dig out some images from there. And we have a collaboration with Rich Mathis that's also looking at simulated Raman scattering and its effect. He's looking at the effects of low-frequency vibrational modes on some chemical reactions occurring actually in our vision in rhodopsin. Okay, so these are the people in the group, and I told them that this is a good picture because kind of they can walk on water kind of thing. I think, and then just I hope you don't mind if I advertise a little bit that we have a possibility for postdocs, and that's what I have to talk to you about. So thank you for your attention, and I'll be glad to get some questions. Yeah.
What kind of background conditions have you operated your system under? Have you tried full daylight conditions or with the lights on in the room or whatever?
Yes. It's a good question. As far as I know, we operate with the lights on, and that is no problem. The advantage that we have is that cars, because if it's a coherent process, you have a lot of things going for you. You know when the signal is coming, and you know more or less where in the spectrum, and in ideal conditions, you know exactly where in space it's going to be. As you go to highly scattering cases where, 100 meters away there's a rock, that's more a little bit more challenging. But you have to take advantage of the timing, the coherence, and knowing more or less in the spectrum. The other thing is, turns out that there's a lot of clever people looking at detection of light, and so there's a lot of clever ways of subtracting backgrounds. So, for example, when we do the selective excitation, it's very easy to subtract, kind of like what changed when you send the correct train of pulses, and you can do that on the fly, so on a single laser shot. So I think that those questions are good. If you would have asked me about this standoff sensing about even 2 or 3 months ago, I would have said, yeah, this is still far-fetched. But we have made some really huge advances, and so I'm beginning to be cautiously optimistic about it. So it's looking very good. Yes. Thank you.
Yeah, I noticed you had Simon T as a collaborator, and he was pretty notorious for poo-pooing all the femtosecond laser applications of cars and saying you need picosecond pulse. I wonder if you turned him around on this issue now.
That's a great question. It's kind of like an insight kind of thing. And let me kind of give you the background. So if I go to my slide number, probably on the order of 3 or 4. Okay, here. Any idea why this is not showing? Oh, yeah, it's there. Okay, so if you remember here, if I take one laser at one frequency and I get a second laser at exactly the right frequency, then I get this beat note, and that actually, if I show you the diagrams that I was showing you before, with the selectivity, this is perfectly selective and there's no other background, and this seems to be ideal. And then you can kind of time the vibrational coherence time in the samples, and it's on the order of 1 or 2 picoseconds. And so there's a very famous Cars microscopist that has been saying, what you need is 2 synchronized picosecond lasers of the right frequency, end of story. Femtosecond lasers will not have any impact in cars, and has really poo-pooed all this idea of using femtosecond lasers. And he's, I wouldn't say he's totally changed, but he is considering that especially the advantage is once you have a broader bandwidth, you can excite, you get a lot more information than matching one particular frequency. And especially for biological samples, there's a lot of richness there. There's proteins and fats and all these other molecules. And so you want a lot more information, and to be selective, you want to be a lot more sophisticated. And the particular collaboration that we have is especially on, I mean, in that case, he's still working around the 3,000 wave numbers, so you still have two different wavelengths, but at least one of them is very broad bandwidth, and the idea is to have, to develop phases that are orthogonal for 3 different types of components. And one is a lipid and one is a protein. And actually, there's 2 types of lipids and one is a protein. And then to come up with the phases and to cycle them very, very fast at megahertz rates so that you can get the real-time microscopic imaging with chemical sensitivity. And that paper is now in review in Nature Photonics. And so I think he's definitely realizing that more bandwidth is better. So I wouldn't quite say that he will recommend femtosecond lasers, but he's getting there. Yeah. Yeah. Yes?
So where do you capture laser there, and the target is the problem,
Okay, it's a good question. Okay. The question is, where is the detector? Now, when you're looking at measurements in the gas phase, ideally, you have the laser, the substance, and you have a detector afterwards, and that's perfect. Okay. Now, standoff detection means that, you know, there might be something explosive down there, and you want to be, you have all these technical terms. You want to be away from the blast zone, which is 50 meters, but much better if you're 100 meters away. And that means that your sensor and any sensing equipment is 100 meters away. And that's much more challenging. And so that means that you have the laser, and now you can use the same telescope for the laser or a separate telescope for the laser and the detection. But everything, you know, we start counting the distance after the last lens. And then there's nothing else out there rather than the target. That makes it more challenging. And what's cool is I convinced the chairman of our department to make us a tube that's 100 meters long next to the lab, so that we can shoot the beam and do some of these experiments. So, and now, you know, I was excited about this. Now, one of the way we work in our group is that I come up with, you know, all sorts of crazy ideas and with fairly high frequency, but I need to wait for students to be excited about it. And finally, now I have an undergrad that's really excited about those experiments. So he's setting up the beam to make it out there. But we've had this tube for a year, and I couldn't get anybody excited from the group. So, you know, so to, you know, when you're a faculty, you need to have the idea, have the student, and have the funding, and then something good happens. And so we're getting there with that. Yeah.
One more question. On that one? No? Okay, so then that's.
Oh, great. Thank you.